For our research interest over in School of Pharmacy, we use H35 and I2 hypoxia workstation. Our research interest in our group includes exploiting the tumor cell energy metabolism in order to improve gynecological cancers treatment. Gynecological cancers in nature are hypoxic solid tumors. That's why we are interested in performing our experiments under hypoxia at different oxygen tensions, whereby we can actually mimic the environmental conditions that we will see in actual tumors. Gynecological cancers are known to have high glycolysis rate and increased lactate concentration due to the reduced oxygen levels, which we call as hypoxia. We know that increased glycolysis rate and lactate concentration does lead to reduction in survival rate and reduction in overall outcome from these patients. Our work using the Don Whitley's hypoxia workstation includes measuring profiling of the gynecological cancer cell lines in order to determine their glycolytic capacity under the treatment of the glycolytic stress test compounds that you can get from Seahorse Biosciences. By using these compounds in combination with our cell lines, we have been able to actually identify possible glycolytic inhibitors which would improve the treatment outcome in these patients. We first seed our cells on plates and then incubate these cells overnight for 24 hours or for the duration of the experiment of the treatment that we are interested in. So the next day the samples are transferred through the transfer tunnel into the I2 chamber. This chamber does provide us the environment that we need to do our analysis by using the Seahorse XF analyzer. The unique thing about I2 is that it has no CO2 in it and it has slightly reduced temperature so that the Seahorse analyzer can function. We also have got an internal incubator which allows us to keep the cells at 37 degrees in a non-CO2 environment at 3% oxygen, which is the oxygen level that you can do your metabolic measurements at, for an hour until the media and the cells are equilibrated to the non-CO2 environment. After this, we then follow the manufacturer's protocol provided the XF Biosciences and do actual metabolic measurements on the cells with and without the glycolytic inhibitors. In future, we're hoping to combine these glycolytic inhibitors that we have identified with the help of the dual hypoxic workstation and the Seahorse XF analyzer with chemotherapy and or radiotherapy in order to improve the treatment outcome for the gynecological cancer patients, especially the ones which has got the tumors that are more resistant to the therapies that are available.